In this video, we'll talk about pathways that reduce NAD+ to NADH. As you recall, NAD and NADH are the primary electron carriers in the cell. NADH takes electrons from food and dumps them off to the electron transport chain during respiration. And the electron transport chain undergoes a series of redox reactions that decrease in free energy and uses the free energy released to pump protons across a membrane, and the proton gradient generates a proton motive force, which is used to power oxidative phosphorylation of ATP by the membrane localized ATP synthase complex. So today we're going to address the question, what are the processes that cells use to transfer electrons from food to NAD+ to make NADH? We'll start by looking at the process from sugars, and then we will take a quick look at how electrons are transferred from proteins and fatty acids. These are catabolic processes. So this entire kind of metabolism where foods are broken down into smaller molecules and are oxidized to extract energy is called catabolism. If we look at the oxidation of glucose to carbon dioxide, here is one way to look at the entire overview. And this is one of my favorite diagrams. It's a free energy diagram showing the three energy pathways that we'll be looking at that take glucose down to CO2. So on the vertical axis, we have change in free energy. So if we start with glucose at 0 here, what you'll see is that in the initial pathway, the first pathway is called glycolysis. So glucose goes into glycolysis, and what happens is that first, 2 ATPs are actually used up. Okay, so energy from 2 ATPs is fed into the reaction to give you a higher energy intermediate. Then this higher energy intermediate is broken down to 2 molecules of pyruvate through a series of 10 reactions involving 10 enzymes. Yeah. And in the process, we get the first extraction of electrons to form NADH. We also get 2 steps that harvest or recoup some of the ATP, and in fact more than recoup the ATP. So we initially use up 2 ATPs, and in glycolysis we make 4 ATPs, so we net 2 ATPs. Okay. And the end product of glycolysis is pyruvate. As you can see in this free energy diagram, going from glucose to pyruvate taps only a small portion of the potential energy that's available from complete oxidation of glucose to carbon dioxide. Okay. So pyruvate still is a reduced molecule, and it has a lot of potential energy still left. And it's only partially oxidized. Then there is a second step called pyruvate oxidation, which is shown just in the shaded area. Okay. So it looks pretty simple. We'll get into it. And this is the initial step in the oxidation of pyruvate. So we again generate more NADH there. And what this doesn't show is that pyruvate is a 3-carbon compound, and the CO2 comes off from pyruvate to generate a 2-carbon compound, acetic acid, which is attached to CoA. And we'll look at that in just a bit more detail later. But the name of this is pyruvate oxidation. Pyruvate oxidation yields acetyl-CoA, and you can see that we're getting lower in free energy now. The acetyl-CoA now enters what we call the citric acid cycle, shown here. And the citric acid cycle completely oxidizes acetyl-CoA to CO2. So the citric acid cycle completes the oxidation of the carbons in glucose to carbon dioxide. And in the process, it nets a lot of free energy, or captures a lot of free energy in the form of reduced electron carriers. So here's NADH, and then here's FADH2, which we haven't talked about, but it is very similar to NADH, and it is an electron carrier. There is also 2 ATP made. So here is the sequence of steps: glycolysis, pyruvate oxidation, citric acid cycle. We'll first take a look at glycolysis. As I said, it takes 10 enzymes to do glycolysis. It's a complex pathway. I don't think there's any point in you learning all of these intermediates or knowing any of the enzyme names. The things that I just want you to know that you can look up the details of the pathway in the textbook or even on Wikipedia. This is a figure from Wikipedia. Yeah. But again, you don't need to know these structures. You don't need to know all of these steps. What you do need to know is what goes into glycolysis. Okay? That the first stages of glycolysis actually use up ATP. Okay? So 2 ATPs. The second half of the reaction nets or creates 4 ATPs, so we net 2 ATPs. And the other thing I want to point out in this figure is the process that makes ATPs on this back half of glycolysis. Okay? This process of making ATPs is that we start with ADP and we take one of these phosphates and add them to ATP to make ATP. This is very different from the oxidative phosphorylation, the process used by ATP synthase. And this kind of a process, the way that cells make ATP by transferring phosphate from one organic molecule to ADP to make ATP, is called substrate level phosphorylation. So again, there are 2 ways that cells can make ATP. One is through oxidative phosphorylation using the ATP synthase and the proton motive force across a membrane. The second way is called substrate level phosphorylation, when a high energy phosphate from an organic molecule is used to phosphorylate ADP to make ATP. Again, the vast majority of ATP in a respiring cell is made by oxidative phosphorylation, but smaller amounts of ATP are also made by substrate level phosphorylation. And glycolysis is one of the pathways where ATP is made by substrate level phosphorylation. This summarizes what I expect you to remember about glycolysis, that it occurs in the cytoplasm of all cells. Unlike the electron transport chain, all these enzymes are soluble enzymes. So initially, we're starting with glucose. The cell uses up 2 ATPs to phosphorylate glucose, and what this does is it is the equivalent of kind of adding activation energy to glucose. You can think of it that way. What then happens is that we have a 6-carbon sugar with 2 phosphates. It's a much higher energy level than glucose. It's less stable. And then the second is to split the sugar into 2 3-carbon sugars. So glucose starts with 6 carbons. You add a couple of phosphates, and then you split the 6-carbon sugar into 2 3-carbon sugars. So glycolysis, the name, says that glyco is sugar and lysis is to break. So glycolysis means breaking glucose or splitting glucose, and splits glucose into 2 3-carbon high energy sugars. These 2 3-carbon sugars then are partially oxidized during the second half of glycolysis. Okay. And the electrons go to NAD plus to make NADH. Okay. So each 3-carbon sugar makes 1 NADH, Starting with glucose, then, we have 2 NADH per glucose molecule. Again, there's substrate level phosphorylation to make ATP, so the back half of the reaction, starting with the 2 3-carbon sugars, makes 4 molecules of ATP. And since we used 2 ATPs at the front part of the reaction, and in the back part of the reaction we make 4, 4 minus 2 gives us a net yield of 2 ATP. Then we have 2 NADH, so reducing power in the form of 2 NADH molecules, and the carbons in glucose come out of glycolysis in the form of 2 pyruvates. So pyruvate is a 3-carbon sugar, which is partially oxidized. When a cell is undergoing respiration, this pyruvate then is oxidized to carbon dioxide plus acetyl-CoA, and it's done by an enzyme complex called pyruvate dehydrogenase. So it has multiple subunits, and pyruvate dehydrogenase carries out 3 separate reactions. Okay? So pyruvic acid is the protonated form of pyruvate. So pyruvate dehydrogenase takes pyruvate, okay, oxidizes pyruvate and transfers the electrons to NAD plus to make NADH. So the oxidation reaction is one reaction. The second reaction is that it decarboxylates pyruvate. So this carbon comes off as carbon dioxide. So that's the second reaction, the decarboxylation of pyruvate. The third reaction is that it takes an organic molecule called coenzyme A and attaches it to what's left, the 2 carbons, which is an acetate. And what you then get is acetyl-CoA, and that's our third reaction. So what goes into pyruvate oxidation is pyruvate, okay, NAD plus, and coenzyme A. So 3 things go into pyruvate oxidation. What comes out are 3 things: CO2, acetyl-CoA, and NADH. So CO2 escapes the cell, leaves the cell. NADH is going to dump off its electrons to the electron transport chain, and acetyl-CoA then enters the Krebs cycle. And this is our summary here of the results of pyruvate oxidation. Now we get to this citric acid cycle. Okay, this is infamous among biochemistry students for being really hairy, and we're going to kind of skip the hairiness, and this diagram summarizes again what I expect you to remember about the citric acid cycle. What goes in is acetyl-CoA, so 2 of the carbons from glucose. Well, actually, we need to multiply this by 2 because per glucose molecule we have 2 pyruvates, and then the 2 pyruvates are both oxidized to acetyl-CoA. So per glucose we have 2 acetyl-CoAs come in. Okay? And then what happens is that we have to multiply everything here by 2 to account for 2 of these things going on, is that the remaining carbons are given off as carbon dioxide. So citric acid cycle completes the oxidation of all of the carbons in glucose to carbon dioxide. In the process, we get lots of reduced electron carriers. I don't need you to remember the exact number. It's nice if you do, but you probably don't need to remember the exact number. Now, it looks here like there's a single step that generates 3 NADH. That's not true. There's 3 separate steps in the citric acid cycle, each of which generates an NADH. But this is just a summary showing you that the various reactions in the citric acid cycle generate 3 NADH. There is, again, a substrate level phosphorylation step. that adds phosphate to ADP to make ATP. Right, and then FADH2 is an electron carrier like NADH. Again, if you ever need to get into the details of the citric acid cycle, you can look it up in your textbook, and you can look it up in Wikipedia. There's all kinds of resources that give you all the details. Okay. So to summarize, we can start with, let's say you eat a pizza. A pizza has carbohydrates in the form of the bread, the crust part. The cheese contains lots of fats. The oil in cheese contains lots of fats. And if you have pepperoni, and even the cheese contains proteins. So we talked about how the carbohydrates, the starch, is going to be broken down into glucose and other sugars, and those are all metabolized through glycolysis, yielding pyruvate, and then the pyruvate goes to acetyl-CoA, acetyl-CoA goes through the Krebs cycle, and then all those NADHs dump their electrons to the electron transport chain. What about fats and proteins? Well, fats are broken down, catabolized into fatty acids and glycerol. Most of that energy in fats are in the long-chain fatty acids. The fatty acids are broken down through a process called beta oxidation. And again, you don't actually need to know the name of that. What you should know is that fatty acids are broken down, and the product of the breakdown of fatty acids is acetyl-CoA. Okay? So there's no energy released during the breaking down of fatty acids into acetyl-CoA, but once you have the acetyl-CoA goes into the citric acid cycle and makes lots of NADH and FADH2, the reduced electron carriers, plus some substrate level phosphorylation to make ATP. Okay? But the main point is that to metabolize fatty acids and get energy, they are broken down to acetyl-CoA, which is then oxidized in the Krebs cycle. Proteins are broken down into individual amino acids, so the peptide bonds are broken to release individual amino acids. Depending on their side chains, some amino acids are converted to pyruvate, other amino acids are converted to acetyl-CoA, and other amino acids are converted to various intermediates in the Krebs cycle. If you're really curious and really want to know how various of the 20 amino acids are broken down or converted into these intermediates, this diagram here tells you some of that. Okay? So some amino acids are alpha-ketoglutarate, fumarate, oxaloacetate, and it doesn't show it, but some other amino acids are converted to pyruvate as well. But again, you don't really need to know. What the point I want to make here is that the amino acids, okay, the earliest they enter into this catabolic pathway is pyruvate. Okay? All the rest are catabolized or oxidized as either acetyl-CoA or Krebs cycle intermediates. Okay? So really, the Krebs cycle is the hub of energy metabolism. And what this figure also shows is that when the Krebs cycle is running, some of the intermediates are used to make amino acids. Others are actually used to go back and make glucose. Others are used to make fatty acids. So if you look at metabolism in the cell, everything pretty much at some point enters the citric acid cycle, or the Krebs cycle is the same as the citric acid cycle, and exit from the citric acid cycle. Okay. And this is a diagram showing in eukaryotes where these energy pathways are. So I forgot to mention that previously all of the enzymes and that we've been talking about, whether we're talking about glycolysis, pyruvate oxidation, and the citric acid cycle, those are all soluble enzymes. Okay? And in a bacterial cell or an archaeal cell, they're all in the cytoplasm. In a eukaryotic cell, these reactions or pathways are compartmentalized. In a eukaryotic cell, glycolysis occurs in the eukaryotic cell cytoplasm. All the rest, including pyruvate oxidation, which is not shown here, occurs in the mitochondrial matrix. Okay? So pyruvate oxidation occurs in the mitochondrial matrix. The citric acid cycle occurs in the mitochondrial matrix. So the pyruvate from glycolysis in a eukaryotic cell has to enter the mitochondrion for anything else to happen. As I said, respiring cells make most of their ATP via oxidative phosphorylation, the ATP synthase here. Okay? So oxidative phosphorylation is done by this enzyme, the ATP synthase complex. Okay? And the ATP synthase complex gets its energy, the power to phosphorylate ADP to make ATP, from the proton gradient across the membrane. Okay? So what happens if there's no respiration? Let's say there's no terminal electron acceptor available. What will happen? Well, if there's no terminal electron acceptor available, the last component in the electron transport chain gets stuck in the reduced form, so it gets stuck holding the electrons. And since it's holding the electrons, all the other upstream components also get stuck holding their electrons. Okay? Then what happens? NADH comes, and there's nothing that it can give its electrons to. So NADH gets stuck holding the electrons. The citric acid cycle, glycolysis, pyruvate oxidation is taking all of the NAD plus in the cell and reducing it to make NADH. What will happen then is that the cell runs out of NAD plus because all of the NAD plus has been converted to NADH. Well, there's other things that happen too. I mean, certainly if there is no electron transport, then proton pumping stops, and as the protons are used to make ATP, the proton gradient will be depleted, ATP synthesis will stop as the proton gradient is depleted. Yeah. So the cell will be in a bad way. It can't make ATP through oxidative phosphorylation. So what can it do? Well, cells have an alternative, okay, to oxidative phosphorylation. Remember, the second way for cells to make ATP is through this process called substrate level phosphorylation. And substrate level phosphorylation occurs during glycolysis. You recall that glycolysis uses 2 ATP, makes 4 ATP, so there's a net creation of 2 ATPs through glycolysis. Okay? But the problem is that in a non-respiring cell, the cell is also converting during glycolysis NAD plus to make NADH. Okay? Now normally in a respiring cell, the NADH will dump its electrons to the electron transport chain so it can be converted back to NAD plus. If it can't do that, if the electron transport chain isn't functioning, okay, the cell has to have another way to regenerate NAD plus from NADH. So to do that, what it takes is the end product of glycolysis, the 2 pyruvates, and it will actually reduce them. It will take the electrons from NADH, give them back to pyruvate, okay, to form a reduced compound. Okay. And in the process, it regenerates, it converts NADH to NAD plus again. Okay. So this seems kind of wasteful. I mean, you used up a lot of energy to make NADH, and now you're expending that energy to go back to NAD plus. But the cell needs to do that if it is to make any ATP at all. So it sacrifices the NADH, but it is still able to make 2 ATP per glucose molecule. And this is called fermentation. Fermentation is the reduction of pyruvate to some other compound, and the entire purpose of fermentation is to regenerate NAD+. Because without NAD+, glycolysis can't go. Without NAD+, none of the other oxidative reactions in the cell can run. And what fermentation does is it generates just enough NAD+ to keep running glycolysis. Okay? So glycolysis and the fermentation reaction, whether the end product of fermentation is ethanol, and this would be in yeast, or in our muscle cells, our muscle cells are also able to run fermentation reactions, and this happens when we exercise so hard that we run an oxygen deficit in our muscles. Then our muscles go into anaerobiosis, in oxygen deficit. They can't run the electron transport chain, they can't run oxidative phosphorylation, so they will actually run glycolysis to continue to make at least some ATP. So they make a lot less ATP through glycolysis and fermentation than they would through oxidative phosphorylation, but at least they're making some ATP. So in our muscles, the pyruvate is reduced to lactate. So again, the point is that the purpose of fermentation is to regenerate NAD+ from NADH, okay, to continue making ATP through glycolysis. Now this doesn't work for either pyruvate oxidation or the citric acid cycle because there is no equivalent reaction to generate to turn over NADHs made from those reactions. So in the absence of respiration, okay, cells can still make ATP through glycolysis and fermentation of glucose. But they make a lot less ATP because if you start with glucose, the net yield is only 2 ATP per glucose. Okay. Compare this to oxidative phosphorylation. If you have in a respiring cell, in oxygen, a cell respiring oxygen, which has the highest energy yield. In a cell respiring oxygen, the net ATP yield, including glycolysis, the substrate level phosphorylation through glycolysis, and substrate level phosphorylation in the citric acid cycle, the net ATP yield, okay, so let me write this down. In respiring cells with oxygen as the terminal electron acceptor, okay, glycolysis yields a net 2 ATPs per glucose. Citric acid cycle also yields 2 ATPs per glucose. And then oxidative phosphorylation yields somewhere between 30 and 32 ATPs per glucose. So the net yield would be somewhere around, let's see, 4 plus 32, 34 to 36 ATPs per glucose in a cell respiring with oxygen as the terminal electron acceptor. Okay. So compare that with just 2 ATPs through glycolysis and fermentation, and you can see why cells have a strong motivation to undergo respiration. Okay. The other thing is that cells cannot run pyruvate oxidation or citric acid cycle, okay, in the absence of respiration, okay, in the absence of a terminal electron acceptor. Since in eukaryotes, The only terminal electron acceptor that mitochondria can use is oxygen. In the absence of oxygen, there is really no mitochondrial function. Okay? So neither pyruvate oxidation nor the citric acid cycle can run in a eukaryotic cell in the absence of oxygen.